This is a special episode of the Stem Cell Podcast, ISSCR 2025 on the ground. Hey everybody, we are Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we're bringing you a very special episode straight from the ISSCR 2025 annual meeting in Hong Kong, which took place from June 11th to the 14th. It was a fantastic meeting. We had the pleasure of talking science with researchers from all over the world. Today, we're going to be bringing you some of those conversations with those brightest minds in the field as we hear from delegates about their thoughts on the meeting. But before we get there,
whether you're looking to attend a conference this year or to expand your network, make the most out of your experience by downloading STEMCELL Technologies' collection of tools to help you prepare for your next event. STEMCELL's downloadable checklists and guides include recommendations on how to get ready before attending conferences, tips for networking, best practices for your LinkedIn profile, and more. Download the conference kit at www.stemcell.com/conference-toolkit. So let's get right into it. As mentioned, Dalen and I attended the ISSCR 2025 annual meeting in Hong Kong, and we had a great time learning about the latest research in the field. We also had the opportunity to chat with attendees and learn about their research. So without further ado, let's introduce some of the attendees that we had the privilege of speaking with at the meeting.
Hi, my name is Daria, and I'm a junior researcher at the Institute of Cytology, Russian Academy of Science, St. Petersburg. So I like cell biology, yes, and we are working on the osteogenic differentiation, how we can either enhance or slow osteogenic differentiation, like or physiological in the bone regeneration, and pathological, like in heart calcification, arterial calcification, and so on. And we focus on the notch signaling pathway because our PI like this pathway, so we should like it too. And also we research interconnection between endothelial cells and osteoblasts. So we co-culture them, and then like in the 3D cultures, and then do RNA sequencing and other things. So I think that I'm working in the science field, not in the pharmacy, because I really want to bring to this world some very important things, maybe a little small puzzle, but I do think that what we are doing, it's very important, and the big pharmacy, it can do things without us. So maybe later I will go to the big pharmacy, but not now. Yes, and talking about the one of the diseases that we are working with is calcification of the arterial valve, and we have a big cardiological center, Almazov Center, in the St. Petersburg, and they replace their valves and give them to us. So this is a very, very huge problem now. And there is no cure, and there's no early markers. So we should really research what is going on there, and not only notch signaling pathway, but other pathway, how they interconnect. We still science know a lot, but these small things, which is very important, we don't know. So.
My name is Hanzhang Ruan, and I'm a postdoc at the Center of Translational Stem Cell Biology, University of Hong Kong. Okay, I'm currently working in the human reproduction field, and I'm studying the maternal-fetal interaction during the early implantation process.
Hi, my name is Xiaoyi Zhang, and I'm a a first-year MPhil student at the University of Hong Kong and also the Center for Translational Stem Cell Biology. So my research is mainly focused on to develop a culture system for the IPS for the expanded potential pluripotent stem cell, which is called EPSCs in our center. And my job is to develop a culture system in compliance with good manufacturing practice standards in preparation for the cell therapy uses. And our EPSC has the expanded potential to differentiate into not only the embryonic lineage, but also the extra embryonic lineage, which is TSCs. And the TSCs can further develop to STB and EVTs.
Hi, my name is Rekha Tripathi. I'm a postdoc researcher at KTH, especially a red postdoc at SciLife Institute in Sweden. I'm a postdoc researcher in Anna Hanlan's group. So in our group, currently I'm working on developing a blood-brain barrier, which is very important for so many purposes, I think, for the drug delivery, for its main focus. But I'm trying to focus around the study on the organoid level, not only focus on blood-brain barrier, CSF BBB barrier. So in current research work, I'm focusing on GLUT1 deficiency syndrome, where the mutation of the glucose gene is which affect the patient and they have epilepsy as a symptoms. So we have a patient-derived iPSCs, and we are trying to model into different organoids. For example, cerebral organoid, choroid plexus organoid, which secrete the CSF, and blood vessels organoid. So the future aim is to understand this mutation in a gene, how it causes, and different phenotype, wide spectrum of phenotype where there is no treatment in a patient. So it's I also got a funding from the SciLife Lab, which is an institute in Sweden, which is collaborative from the KTH, Stockholm University, and Karolinska Institute. So in Anna Hanlan's lab, we also have the organ-on-chip model. So it's a very nice combination where we try to do the organoid on a chip for drug screening. So overall, I'm trying to focus on to unlock a brain barrier mechanism for a future drug screening.
Hi, my name is Matthew Chang, and I'm a PhD student at UHN in Toronto. Yeah, so I study human heart development, and then I'm using a couple of the new embryoid models. So I'm using specifically the gastroid model. And what we're interested in doing is sort of understanding more the morphogenic processes of cardiogenesis, like the formation of like the cardiac crescent and why that happens and how did those progenitors end up there and stuff like that. Yeah, it's been a little challenging, I'm not gonna lie. I think just because the nature of like the human gastroid system and some of the human embryoid systems are quite new. One of the biggest challenges I have is like the reproducibility issues. So I find that they're not that reproducible, and then that just makes a lot of the downstream analysis and experiments a little bit more difficult. Yeah, my lab, like I'm from Stephanie Protz's lab, okay. And like we do a lot of directed differentiations into all the different subtypes of cardiomyocytes, and then we're really interested in doing like transplantation and more like clinical use. And then I'm the only developmental biologist there.
So hi, my name is Christian Roigelboy. I am a graduate student at the University of Cincinnati and Cincinnati's Children's Medical Center. Yeah. So I really study pregnancy hormone and its role in development in the gut. I really am passionate about this because working in Cincinnati's children, I'm a firm believer that before we can take care of children and illnesses and diseases, we have to take care of the mothers-to-be. So if we understand how hormonal, especially in pregnancy, affect gut development, we can better women in getting nutrition adapted to them, to their plans specified for their childbirth. We can help prevent and minimize postnatal diseases and chronic illnesses in children. And we can also, adapt these plans and these research to second and third world countries with mothers that often do not get the nourishment that they need.
Hi, my name is Eric Harris, and I'm a research assistant and part-time PhD student at the University of Leeds. So, unlike a lot of people, I'm studying rare neurological diseases, so really rare. So, one of the diseases I study doesn't even have a name, because there's like 12 people in the whole world who've got these phenotypes. And we're trying to model these really rare neurodevelopmental disorders. They're all mostly characterized by macrocephaly or microcephaly, and they have other overgrowth defects like polydactyly, so extra fingers, extra toes, and heart, kidney problems, etc. But we're trying to essentially use iPSC models to model these rare disorders, because there's not that many people that model the rare disorders in these formats. So one of them doesn't, that we're studying it. We're calling it max-associated global development disorder at the moment, because originally 3 patients have this gene. It's a lot of jargon, isn't it? But then now more patients are coming forward with that mutation, and so when there's enough patients with it, it will have a name, I'm sure. So yeah, my PI also works is more as a geneticist as well, so that's why they work hand in hand, that sort of thing. But I do work on, I say, more common rare disorders. So Alexander disease is an example of a leukodystrophy, a white matter disorder, and I think the prevalence of that is like 1 in 100,000 or something like that, but still very, very rare, but not quite as rare as 12 people in the world. So,
hi, my name is Tomáš Gerecsei, and I'm a sales manager at Cytosearch. Yeah, of course. So at Cytosearch, we are working on fluidic force microscopy, and that technology as a whole has many different aspects. So the technology started more from biophysics, which is also where I started my career from. But right now we have a lot of applications in more like molecular biology, which is also why we are here at ISSCR. And most importantly, we do sequencing, a very special kind of sequencing where you don't have to kill the cell. So of course, everybody knows that for RNA-seq, the first step of the process is you lyse the cells. And that, of course, is the last thing that the cell will do in its entire life. And that's a great assay, but it's an endpoint measurement, very inherently. And that is a big limitation for anyone who's looking at dynamic processes, which of course in stem cell research, that's all around us, because differentiation and reprogramming, that's all happens in time, right? And with normal RNA-seq, that is really difficult to capture in spite of developments in computational techniques and all that. And so our approach is to, instead of destroying the whole cell, we take what we call a cytoplasmic biopsy. And that's very similar to biopsy. When you go to the hospital, they don't take all your blood to do a check. They just take a tiny sample and then they do all the checks on that. And then you still survive and you can go on about your life and do all the things. And it's the very same idea for us. So we take a biopsy from the cytoplasm, we do the sequencing or even eventually proteomics, and then the cell is still in your sample. It's possible to follow it, differentiate it, do any kind of regeneration or whatever protocol, and then see what it does, while at the same time already having the transcriptome in your hand. So it's a very similar idea to, you go to the doctor, they take your cholesterol, and then they tell you it's too high, so then you start to eat healthier, and then they can check it later again. Did that do something to your molecular characteristics? And we can do the same. So we take the RNA-seq, we can differentiate the cell, and then later on you take another RNA-seq from the very same cell, and then you're able to see what happened. So that's the idea.
Hi, my name is Luc van Heugden, and I'm a master student at the University of Zurich. So originally I was very interested in cancer research, but I had a course then in stem cell research and in tissue engineering, and that sparked my interest then. And that's why I'm here also at the convention, to get an insight into all the topics for what I can do for my master's or what I can do in the future, even afterwards for my PhD. And up until now, it's amazing what possibilities you have with tissue engineering and differentiating in any cell types.
Hi, I'm Nina Horowitz, and I'm the CEO of ImmuneBridge. So ImmuneBridge is a cell therapy development platform. We've solved the 2 hardest problems in cell therapy, which is finding good donors and then manufacturing them at large scale. So we enable anybody to make a high quality therapeutic at a few $1,000s a dose at most without relying on iPSCs. And what do we do? So we have an HSC expansion molecule called IBR403. The reason that's special is it's actually really hard to get HSCs to proliferate, but also maintain their multipotency. you can typically only do one or the other. So we found this molecule that does both, meaning we can expand them 1,000-fold up to 100,000-fold, and then turn them into any kind of immune cell, NK cells, T cells, macrophages, neutrophils, whole nine yard. So because we have this platform, but we're just a tiny company, we can't make all of those therapies ourselves. So we work with academics, small biotech, big pharma, to enable them to develop whatever cell therapies they want on our platform. My goal is to have 10 therapies in the clinic in the next 10 years.
Hi, my name is Sina Bartveld, and I'm a professor for medical biotechnology at the Technical University Berlin. So we work with adult stem cell-derived organoids, so mainly of the gastrointestinal tract, only human. And we use these organoids as models for infection biology. And it's super important because there's this huge field of infection biologists, and they really lack models. There are so many bacteria and viruses that infect us humans specifically, and they do not naturally replicate in mice. And so a mouse model is not always the best version to analyze such bacteria or viruses. And this field really lacked very good model systems. So organoids provide this new tool. And I originally came from the field of infection biology. And then I joined the stem cell field to actually make the human gastric organoids and infect them with Helicobacter pylori. And that kind of skyrocketed a bit. And now a lot of infection biologists are using organoids. And that's really great to see.
Hi, my name is Ruben de Vries, and I'm a PhD student at Micha Drucker's lab at the Leiden Academic Center for Drug Research in the Netherlands. I'm working on spinal cord development and how neural and glial cells interact with each other, and how they might also need each other during development to reach this final functional endpoint where we can generate a spinal cord in a dish model. So our lab is working on different approaches to do this. And I'm specifically looking at the dorsal root ganglion, so sensory neurons and their glial counterpart, the satellite glial cells, and the Schwann cells that also make up this structure.
Thanks to all these attendees for participating in this episode. And there's definitely some really interesting research areas here that we'd love to talk about more in detail in future episodes.
Yeah, man, I agree. It's my favorite part of every year when we do these ISSCR on the ground recordings, getting to talk to a whole range of attendees from, trainees, people just starting their doctoral studies, all the way up to, professors and CEOs. It was really one of the best things about this year's conference for me, and so much fun.
Yeah, a ton of fun. And, we also wanted to get a sense of what folks were really most excited about at the meeting, beyond just the dim sum, or, what they were most looking forward to seeing. So let's hear what they had to say.
Hi, it's Daria. Yes, the talks, I think, because my poster session will be at 4. And my colleague, Anna, she already have her poster session. And not only she tried to speak English, I'm very proud of her, but we now have a small problem. We can't differentiate iPSC cells into MSC cells. They just die. And we tried to find new connections, maybe collaborators that can help us, and maybe we can help them. And we found from France, beautiful people, I think, and yes, they will give us protocols, and they don't know how to differentiate iPSC to the endothelial cells. And my colleague do it every day. So it's really nice. Because it was a really huge problem in Russia, because we don't know who do it, who not, and here we can make connection with the Americans, with the old people, and it's really nice.
Hi, it's Hanjang. Actually, this is the first international conference I have attended. Because of the pandemic situation, I didn't go anywhere during my PhD studies. So this is the first time I see so many people come from all over the world and talk about the science. And I can see, because this is a very broad conference and people are studying in many different fields. And previously, I only went to the conference that focused on the reproduction or the human stem cells for the reproduction studies. So I just realized there are so many other studies using the stem cells and can do very cool things and use very different animal models, even like the very small organism for the studies. So very fantastic, and it's kind of also inspiring me. Like we can study the reproduction maybe from the revolution aspect, but not only just focus on human ourselves. And also there are many famous people coming. Yeah, and like the many one of the author of the human blastoid, author Nicholas, we have been following his study since like 2022 when he published the paper of human blastoid, and we try to follow his protocol to repeat his experiments and for our study. And it's quite cool to see a real person in front of myself and know what he's doing now by using his model. And yeah, hopefully I can take a picture with him afterwards during the poster session if I can catch him.
Hi, it's Anna. Before I came to ISSCR, I have never expected to see so much studies about iPSCs in advance, and they have many advanced applications to cure various diseases. And yeah, I will say ISSCR opens my eyes and broadened my view of the iPSC research and the potential applications. Yeah, and I also see many potential problems of the iPSC uses because people seems to be too urged to apply the iPSCs into the cell therapies. So there are many discussions about the regulations to see whether it is appropriate to directly use these ATPs into the cell therapy applications.
Hi, it's Rekha. Okay, so the obviously Hong Kong. That was one of the more exciting part because I never been here, and it is one of the beautiful country to be here. You know, like city wise and a whole experience of shopping. And I think more than me, my friends were more excited that I'm going here actually, and they couldn't actually. So they kind of envy me. So I being here, and more than that, I think this was my first conference I have attended. I never attended it. And now I understand why people talk about this. Therefore, if you are in a stem cell research, you should join this. You should definitely attend this. And it's worth it because you have a very fantastic speaker, I think. And the poster presentation of various very different topics, which can you give a glimpse of overall research, what is happening, which is very important, I think, because when we are focusing, for example, I'm focusing on brain, probably I'm going to be like very narrow focused on the words and I don't know what is exactly happening, but it can be applicable for me in a future way. And I'm a postdoc researcher. So networking, meeting a very motivated people and the PI, like the professor, and keep on exciting as being scientists. It's one of that feeling actually I got it. So which is really nice because as a researcher, you may have a motivation feeling up and down where you start questioning why you are doing it at first place. So I think this kind of venture, like this kind of exciting conference really motivate, it's valuable research. I think so. I think it was really nice to be here.
Hi, it's Matthew. So I've been to Hong Kong a couple of times before, and like my favorite part about Hong Kong is like it's full of character because one, like I love food, so there's a lot of good food around. And then in terms of like places to be, like I think Hong Kong has a big amount of diversity. And then also like ISSCR, it's like I was really interested in meeting different people who work with stem cells. It's like my first ISSCR, and it's been a pretty big experience for me.
Hi, it's Christian. I was really excited to come to an environment that was full of basic science research that focused on stem cell. I think stem cell is a topic that is getting really hot, and it's great to be in a community with other graduate students from all around the world, different nationalities, different backgrounds, to come together and share their science on stem cell work.
Hi, it's Erica. I was really excited to meet more people who do the same sort of models as me, because in my institute, I am the only one making iPSC-derived neuroprogenitor cells, forebrain neurons, astrocytes at the moment. And I work alongside like people who do like kidneys and retinas and things, but I'm the only one doing it in my team at the moment. Not as in wide of the university, but we're like a smaller institute. And so I was really looking forward to sort of meeting people and talking about the problems that I have and hoping to get some help, really. And I think I have, so that's great.
Hi, it's Tomasz. Yeah, I was just wanting to see where is the whole stem cell field right now, because I personally am not so experienced in this field. And I just wanted to know what the main conversation drivers are right now. And for that, I think this is the best conference, of course, because you have all the brightest mind in this field. And I think it's amazing, the progress that is being done, that now we are even have to have a separate session about the ethics of everything. Can you grow humans in a dish and all that? So yeah, this field is definitely something to watch, that I think everybody should kind of keep an eye on this.
Hi, it's Luc. Yes, it was for the networking. I mean, I knew that I wanted to start early with networking and connecting with people, getting to know people, experts in the field. And actually, I was targeting CIRA people themselves specifically because the University of Zurich has a strategic partnership with CIRA. And they proposed to me that I would go for an internship in Japan. So I was definitely trying to find these people there to know them beforehand.
Hi, it's Nina. So I actually just came from a natural killer cell conference in Tasmania. Like literally was at 4 days of the conference, got on a plane, slept, came here, and then came here and started coming to the talks. And I think it's really easy sometimes to get lost in the world of one cell type because there's so much going on just in the field of NK cells. And I think that's like where my PhD experience is and where a lot of my coworkers' expertise lies. And so I was really excited to come here and just zoom out and like get back to the big picture and think about what are people doing with other immune cells. Beyond that, what are they doing with MSCs? Beyond that, what are they doing with iPSC-derived embryos, things like that. And I actually ran into one of my grad school professors, Bo Wang. He gave a great talk on flatworms and how they can live forever because they can regenerate. And like, we don't even think about tissue regeneration, but a lot of the pathways are actually really similar. So it was great to come here, zoom out, get my perspective broadened again, and then see what we can learn from these other fields and how we can bring that back and implement it in our platform.
Hi, it's Sina. Well, of course, you're always excited to see new science. But to be honest, at this stage, it's also a lot of about meeting friends and also making new friends, like meeting people whose names you've heard, whose research you've seen, but you haven't met them. And I was also really excited to go to Hong Kong because there is a person here. She works also with human gastric organoids, and we met some, 12, 13 years ago. And it was just also great on that personal level just to meet up with her and with two friends, actually. And that was really good. So meeting scientific friends is great and making new scientific friends.
Hi, it's Ruben. And I was really looking forward to seeing all of the applications that are finally coming to a final conclusion, let's say. Well, maybe not final conclusion, but to a sort of an interim conclusion where the feasibility of a lot of these stem cell therapies, ex vivo gene therapies, are finally being applied and not just being tinkered with in the lab. I think that's also, it helps me stay motivated doing the fundamental research, tinkering with stem cells in the cell culture lab, seeing that 20 years down the line, maybe these things can be used for an actual application, or maybe sooner. But I came into science maybe a bit indirectly. Both my parents are doctors, so I always, from a young age, had an interest in sort of the life sciences, but they never really pushed me in this direction. They just sort of came along with the upbringing. And then when I went into university, I very quickly realized that this curiosity-driven work is really for me. And I also love the freedom and creativity that I get in this work, in this cool developing field, to really follow what I'm interested in.
Yeah, as you heard there was a lot covered at this meeting, diverse research areas and speakers and some really exciting talks. One of the things I was most excited about was our live episode with Drs. Han-Ki Dang and Candice Liu, who shared about their groundbreaking studies in chemically induced pluripotent stem cells to treat type 1 diabetes. You guys got to go back and listen to that one if you haven't already. It's episode 297. And the lineup, so many sessions to choose from this year, from the many different, not just talks, but sessions on kind of the bioethics. I mean, not just technical science talks, but about the bioethics, a lot of methodology, great stories told all around. We were running around like crazy, but we wanted to hear which sessions the delegates found most interesting. So let's hear what they had to say.
Hi, it's Daria from the Harvard, I think, about the brain organoids, and they used to do it like for five years, and then they started doing it like for 2 weeks. Oh my God! And the methods they use, it's huge. I really like, and the images of the brain signals. I used to research in the neuro, but transformed to the cardio, it's okay. And it really impressed me.
Hi, it's Hanjang. Yeah, it's not about the reproduction, but it's about the brain development. They found a gene that enriched in human, higher expression in human than other animal like rabbit or some small animals. And they found that knock out this gene, the brain organoids will grow smaller. So they think this gene helps human to get a larger brain. That's quite cool, I think. Yeah, very impressive. We actually can just have one gene, that master gene, that help us to be different from other species.
Hi, it's Anna. I would say the most recent I could recall is the one seminar I heard this afternoon given by Dr. Nicholas, and he was talking about his research about the blastoid models and in combination with the trophoblast to see the embryonic embryo invasion, and they even found that there was a virus infected in the old world monkeys.
Hi, it's Rekha. I think first Pascal, he's like one. I think for when it comes to organoid, I know him quite much research paper focusing on the brain organoid. I think he always been a motivation factor for me because I'm in a brain organoid research. So that has been very nice. And I think quite many also early research talk were also nice, where they are focusing on very different models to be created in the organoid, which I haven't actually thought about it. For example, kidney model. I don't remember the name, unfortunately, but I do remember the topic. kidney organoid or the vascularization, or like especially single cell method, which they have been using for the embryogenesis or something. So it's really nice. And actually it's a two-way that to see how to present the research in an effective way, because it's like learning in a both side, not only topic and how to, you should have your slides, how to present it nicely, and what content you should have to bigger audience, because the audience is going to have a very different level of knowledge. So I think that is also, I take home message kind of like, how to prepare yourself actually for this kind of conferences.
Hi, it's Matthew. Yeah, honestly, it was to like see if there were a lot of people doing like some of the gastroid models and some of the early embryoid models, because like I think it's like over the past like 2 to 3 years or something, there's been this like huge explosion of all the different models. So it's always nice to like talk to the people who've like discovered or who use these models, and then talk about like why they use them or why might they not have used them or they've moved away from it. And it's like really helpful for reinforcing my experiences, because like I don't know that many people like who work with the gastroid model, and like I was having a lot of issues with reproducibility, and then like just talking to a bunch of people who work with it here, it's like they're also experiencing the same things. So it's nice to have that like common bond. Yeah, so I've seen like Jakob Hanna talk a couple of times. Like there was a conference in Japan that I went to like 2 or 3 years ago, and he presented like the human synthetic embryo model stuff before then. So it was also really interesting just to see like what he was doing next. And yeah, his AC media seems pretty cool. I think it was the first day. There was the I think it's the Allen Institute talks for imaging. So I also really like microscopy. So it was a really cool session, and some of the tools that they're using. And like I think what's really interesting for me is like how do you take like microscopy images and stuff like that and like get meaningful data out of it. And it was such a cool experience just to hear all their different algorithms that they're working on, all their different model systems, and like how they're doing. Like one of the coolest things was I think it was the agave reconstruction, and like the images and the renderings that they got out of that were like so beautiful.
Hi, it's Christian. Yeah, so it was actually on day 2. So yesterday, it was to talk about regeneration of the flatfish model, that if you cut off his head, the body was able to regenerate through what we believe is the MAP-ERK kinase pathway. I found that really interesting because it's very similar to the model that I'm looking at with HCG, which is one of the first pregnancy hormone that primes the gut to regenerate and prime the body for pregnancy. So it was really interesting of the theory that I believe HCG acts upon is the same model and pathway that this fish uses to regenerate its body and its stem cell population. It's also a really cute fish. It's great science, but also has a cute model. So it's a win-win in my book.
Hi, it's Erica. It's a really hard one. So many different ones. I would say from a just mind-blowing perspective, I've forgotten the name. I'm going to have to go back and find it. But there was a talk on essentially assembloids and how I've read about sort of using like 2 different kinds of organoids and putting them together. But I've not actually seen 4 put together, which is mind-blowing. And actually to see how you get this periphery like to the cortex sort of circuit actually in an organoid is crazy to me. Even as someone who works in this field, I still sort of blink and go, that can be done in a Petri dish? Seriously? That's amazing. So yeah, I think that was mind-blowing. And it was very much like feel like an imposter syndrome in this room because I'm not doing anywhere near that sort of thing. And it's amazing, really.
Hi, it's Tomasz. Yeah, so this is something that, of course, you see in like Black Mirror episodes more, or, you know, like crazy movies. And it's really interesting to kind of consider that, of course, this is very important research because it will help you at the end to develop medicines to make people's lives better. But it really makes you think about, you know, just life in itself, which might be getting a bit out of hand, the conversation. But the point is that, yeah, like it makes you think, you know, when does the life begin? How long can you grow an embryo when you're still trying to help people? But, you know, these technologies in the wrong hands maybe can be a bit crazy.
Hi, it's Luc. I don't know the name, unfortunately, but I really liked the spinal cord injury regenerative models, especially with zebrafish. That really inspired me. And I also can remember the talk about the brain organoids from Paulo Arlota in the Grand Hall. That was also really amazing. And I also found very easy to understand and very good to follow the talk with thymus regeneration. Like I'm very interested in the clinical approach and the translation of this research that we do here into then clinical aspects.
Hi, it's Nina. I feel like it's so hard to choose because there's a bunch of them. I'm going to shout out Lily Yang, actually. So she is a professor. She gave a talk on making NKT cells, which have some benefits of natural killer cells and some benefits of T cells. And they do this really unique targeting where they can kill tumor cells, but also within the tumor microenvironment, they'll kill the suppressive immune cells, like the myeloid-derived suppressor cells and the M2 macrophages, but they won't kill the cytotoxic CD4 and the helper cytotoxic CD8 and the helper CD4 T cells, the ones that you want to mount an immune response against the tumor. And I was like, that is the coolest multi-targeted therapeutic I've ever seen. I went up to her right afterwards. I was like, we want to help you manufacture this. The fact that you can go in and selectively turn off only the bad parts of the immune system, but not the good parts, is, I think, a huge benefit.
Hi, it's Sina. Well, I really like the meeting as well, because on one hand, it gives you this broad perspective. Although I'm only in the gastrointestinal tract, it's super cool to see all these brain organoids. And I think this year it really stood out that there's a lot of embryo models around and a lot of more integration of these ethical aspects of the brain organoids, but as well of the embryo models. And that was really interesting. But of course, I was very much looking forward to see my topic. So the use of stem cells and stem cell-derived models for infection biology. And here the meeting had a special section on that. And I was really looking forward to see that. And I enjoyed it a lot. It was also organoids, especially respiratory organoids for viral infection, and then also some organ-on-chip technologies, which is like the next cool model, and how to make these organoids more complex. And it's very interesting. Yeah, I liked it a lot.
Hi, it's Ruben. I mean, the one talk that really, I really loved the talk with Deng about the chemical reprogramming and the application showing that it actually works for the treatment of diabetes. But I also really loved the talk by Hideyuki Okano that was working on the spinal cord injury transplantation. That's also a bit closer to my field of interest. And him showing these things is remarkable. I mean, I was really blown away.
Definitely some great responses there. I felt the same as many of the attendees who shared their thoughts on the talks. I was in the room with them in many cases. Arun, what was your favorite session?
You know, I really liked that session focused on chimeras and these next generation organoid systems. Really incredible work happening in the human animal chimera area. Of course, spearheaded by the work that was done by Hiromitsu Nakauchi almost 15 years ago now. But now you have other folks in the stem cell field and all across the world who are trying to improve the efficiency of these chimerism models. That's just a really exciting area of study for me.
For me, I mean, this is weird, but it was more the experience, the way I felt during the talk. There were so many to pick from in terms of the excellence and the the awe that I felt in watching them, and whether it was like 5-year-old organoids and beyond from Paula Arlata, or seeing Jacob Hanna extend his ex utero embryo culture device out to the point where these mice fetuses were forming limb buds and even digits. So a lot to see that was crazy and a feast for the eyes and the mind. But for me, the experience that brought me back was hearing this study about the green anal lizard, right? And the reason I loved it is because hearing the enthusiasm of the investigator talk about creating the tool set to look into this, and just totally from space, really took me back to the discovery you feel when you're tinkering. So I really enjoyed that, and enjoyed the emphasis that the organizers placed here on getting investigators that had not presented at the ISSCR before. So that was really great, but really too many to choose from for me. You can catch up on all the sessions we attended by listening to our daily episodes from the conference if you haven't already. We got some more conversations with delegates in just a bit, but before we get to that,
do you want to implement the new ISSCR standards for human stem cell use in research? Get a free checklist wall chart that helps you follow the recommended reporting practices and quickly reference the relevant sections of the standards. To learn more, visit stemcell.com/ISSCR-standard/wall chart. So another episode we want to mention is that back in March, we were really fortunate enough to chat with Drs. Valentina Greco, Catherine Shea, and Eugenia Pedini about what they were looking forward to at ISSCR 2025. And, one thing that Dalen and I, as well as the ISSCR leadership, what we were really excited about was getting to explore Hong Kong, of course. I mean, eat everything in sight for me, for sure. Everybody pretty much mentioned the food, which was, of course, great. But we wanted to hear about the attendees' adventures outside of the conference center as well. So let's hear what they had to say.
Hi, it's Daria. I said that before the podcast that I really like Asia. I like the culture. I like the food. I like nature. I think that in my future plans, I would like to live in Thailand. And I've been in Bali too, and I'm like comparing these countries. And I really like Hong Kong. I don't like the rain a lot, but in St. Petersburg we have a lot of rain, like in London, so it's okay. But very beautiful. I like the fact that lots of people speak English, and you can go to every person on the street, and they can speak to you a little bit or with gestures, but you understand each other. So it's thank you for my PI that we have this great opportunity to go there and see this beautiful city.
Hi, it's Hanjang. I have lived in Hong Kong almost 7 years because I finished my PhD here, and I also work here afterwards. And I think the big difference between Hong Kong and the US and UK is that Hong Kong have so many delicious food here. Yeah, I like the fish and chips and I also like hamburgers, but still I like Chinese food. That's my favorite thing. Okay, and also Hong Kong is like other cities, it's still very international and so many opportunities here. I think in Hong Kong, they support everything that you are trying to achieve. They give, like the University of Hong Kong, they give you many support and help when you're doing your research. There are many centers, but not only focus on the research, but give the service during the research. When I study in the University of Maryland, they have the renting center also helped me a lot. Yeah. So I think because I think still I'm Asia, I'm a Chinese, so I'm more like the Hong Kong is my favorite city.
Hi, it's Anna. Yeah, definitely. I love Hong Kong. The first time when I came Hong Kong was 6 years ago, and the super city of Hong Kong gives me the impressions that people are always busy and the buildings are crowded. It seems so, but with my time of studying here and living in Hong Kong, I have more chances to get in touch with the people in Hong Kong here. And I was impressed by their enthusiasm and their kindness to help people in need. But the foods, at the beginning, I didn't really get familiar with it because the food flavor in our hometown is totally different from here. So yeah, and also one more thing that attracted me most about Hong Kong is that I think Hong Kong is a city that allows people to live friendly with the animals and the environments, and people here are very they have the conscience to protect environments, and I think even the IPS grows so fast, the industry goes so fast, we still need to think about how to be with the environment friendly.
Hi, it's Rekha. It makes me feel home. It may sound weird, but I'm actually from India, from Pune. It's similarly, you see super crowd, number of people around, happy, like being, if you ask help, you are getting help, you know. So because I live also in Mumbai, the weather is kind of similar, humid. So it's quite many things. I think it's more similar to my home. So it made me feel home, actually. And the food, I really like it. So I'm a big fan of dumplings and other food, seafood. So for me, it being actually really nice. So India, I will go in December. So I'm not missing anything in the summer at least. You may gonna say that I'm a man, but I really like rain. Because I come from Pune where we have a monsoon season like crazy. So I like rain. I don't have any complaint, actually.
Hi, it's Christian. I mean, apart from the jet lag from coming from the US, I mean, Hong Kong is phenomenal. I think one of the really amazing thing that has really stuck out is the kindness and the generosity of the people here in Hong Kong. They have been super welcoming. I mean, apart from the food that's delicious and I can eat noodles and pork all day, every day if I could. It's really the personality, the friendliness, and the welcomingness of the people of Hong Kong that has made this experience, both inside at the conference and outside of the conference, a really amazing experience trip that I'm very honored to have been part of.
Hi, it's Erica. The food, definitely. I mean, it's quite hard actually though as a vegetarian to find food, but I have really enjoyed the food and the hustle and bustle of it all. I'm from the UK, but I'm not really a big fan of London. And it's very different to London, I think. Like it just all makes sense. I like it a lot here. I haven't had much chance to go and do much touristy stuff yet. I'm going to go to Victoria Peak tomorrow, obviously. That's got to be done. But no, I like it. And I like how clean the subway is, or the metro, or whatever they call it. Because again, in London, it's really not that clean. Again, here, like people are so kind and really happy to help you, which I love. I love that there's people dotted around everywhere, almost like it's their job to just help you. They're not. They're just going about their day as a person who lives in Hong Kong.
Hi, it's Tomasz. So I'm very, very lucky because I'm here with our amazing Chinese distributor partners. And as always, they are the amazing hosts. And for me, whenever I come to China, the highlight is always the food. So obviously, Hong Kong is no different. You know, Cantonese food, very special stuff. And my highlight was definitely the roasted goose, which, I don't think I had ever a more delicious bird on my plate.
Hi, it's Luc. I mean, I haven't seen much of the city yet, but I definitely like the food. I like Asian food overall. And I was with my friends in local cuisine, like very local restaurants where normally the tourists wouldn't go. And I really like the seafood from these restaurants. But sometimes it's difficult with the language barrier because you cannot read the card. It's all in Chinese. I don't understand the letters. You don't see the pricing or what you get afterwards. It's always a surprise.
Hi, it's Nina. I like the city feel. I think it's a very different feeling city than San Francisco or Boston. It feels like powerful. It has a really good energy. So last night we were at the Ozone Bar, which is the highest rooftop bar anywhere in the world. And we were just looking out at the city. It It looks so beautiful. It's got all these amazing lights. I'm doing a boat tour tomorrow to see the light show. I like the hustle and bustle in the sense that it's like as busy as New York City, but it feels cleaner and safer and more fun, and the food is really good. Not that I don't love New York. The bagels and the pizza are undefeated, but I've really been enjoying just experiencing a different culture, honestly. Yeah.
Hi, it's Sina. Just that it is so different. And I mean, like you guys, I've been jet lagged very much. And I didn't also, I came in here very briefly, so I didn't have time to do any much touristy stuff. But just to see everything being so different than at home, it's very very full and just buzzing, and the skyline is amazing. And seeing as coming from Hamburg, I always seeing a river with some boats floating through always calms me a lot. I love that. So that I just really like it, and I've been taking a lot of pictures and little movies and sent them to my kids, and they're all in awe. So we have to come back.
Hi, it's Ruben. I mean, this city is impressive on all sides. Like just arriving here, taking the train from the airport, and that first view of the skyline. And then, I mean, I was here a few days early, so I had the chance to really explore a little bit, see the life and the busyness of the city, enjoy some of the good food. I think that's what everyone comes to do in Hong Kong when they get here.
Yeah, for sure. There's a ton to do in Hong Kong. It's such a beautiful city. It's more than a skyline, as someone has said before. It sounded like everybody just had a blast, just a great time. I mean, for me, of course, it's the food first and foremost. I ate everything in sight, but also getting the chance to explore the sights of the city and going up to Victoria Peak the Tuesday before the conference and getting a view of that skyline from the very top. That was gorgeous, and I'm glad I got to do that before all the rain set in.
Yeah, for me, I was blessed to have my dear wife along on the trip with me. So she set a lovely itinerary. But mostly what I did was sweat, the humidity. And if I was outside, I was sweating. But I will give credit to Hong Kong as a city. The design is amazing. All these elevated walkways, air conditioning is generous. I heard about how they went Rambo with the AC here. Now I understand why. But I would say the highlight for me was Soho. I didn't know there's a Soho in Hong Kong, which really reminded me of the Soho of my native New York, and the mix of so many cultures, a lot of Western, I'll admit, but the mashup and the fusion and the smells, honestly, in the street, the combination of the I don't know, dim sum-ish flavor with kind of that cheesy-ish flavor you get walking through, around a butcher shop or whatnot. It was a real feast for the senses. And the people were so cool and nice and well behaved. Last time I was in the subway in New York, no joke, a guy was flipping out, screaming at the top of his lungs. I cannot imagine that happening on the subways here. Honestly, weirdly, my favorite part was riding the subway in Hong Kong. So there's that. But I have to agree with all the attendees and their answers. Check, check, check on every one. Cannot disagree. So much to love in this town. Well, that brings us to the end of our ISSCR 2025 special. We had a great time at the meeting and hope that you enjoyed hearing from some of the attendees. Don't forget to check out all the episodes that we released during the meeting. You can find them at stemcellpodcast.com or wherever you get your podcasts. Until you hear from us again, thank you so much for listening.